So thanks, Mark, for joining us today. For the audience, Mark is specializing in engineering rapid prototype of software for multidisciplinary data analysis project. He started his training at community college level and achieving his AA and completing his master's. His experience is in bioinformatics, a crossroad of computer science and biology. So I'm delighted to turn the podium over to Mark to talk about what he's up to and what he's doing at this crossroad. Thank you,
Th-
Mark.
Thanks, Azad. Appreciate that introduction. And like he said, I started off at the community college level and went on to getting my bachelor's and then master's. So, I really think the community college level is very important. All right. So with that, might as well jump right into the talk. So the title is Bioinformatics, a crossroad of computer science and biology. And if that doesn't get you excited, don't know what will. Um, if there was a live audience, I would ask if who all knows about bioinformatics. Izad, had you heard about bioinformatics before?
Yes, I have, and it's actually a very very exciting field. As a patient who is using those services,
Right.
a lot of the bioinformatics is used to predict, especially with gene and gene therapy coming online. Your work is extremely important because of the data analysis and data science you do.
Right. So right, that's awesome. And you're a pretty informed person on that. Most people hear it and think, oh, what is that, right? So kind of get into the we'll get into the let's see. We'll get into the kind of how the talk's going to go and kind of, elucidate what bioinformatics is. Izad obviously knows, but there'd be a lot of people that have no idea what bioinformatics is. So going to start by giving a kind of a brief history of computer science in general and kind of how it ties into bioinformatics, starting in around like the 40s or 50s, right there around World War II. Talk about what is it, how can you use it, how can you learn it. I'll talk about my experience, how I gained my training through like you already said, community college, undergrad, and then graduate school. And then we'll kind of end with talking about a few projects that I think highlight the crossroad. Like that's the whole point of the title, right? Is the crossroad between biology and computer science. So the projects I'm highlighting are really showing like how things that you might not think in computer science can also be applicable to biological problems. So I was going to ask people if they knew who any of these people were, but Azad might recognize a few of these guys. So it's back in the day when computers weren't very glamorous to use, right? They're just used to kind of tabulate, polynomials like x cubed plus 2x, things like that in large quantities, and a lot of times. This is Charles Babbage, who, he made the analytical engine and the difference engine. This is it right here. You can see you kind of click those things to put in your equations, and then it'll tabulate the answers for you, which is pretty cool. I've never used one, but I've always wanted to see one in person. And then obviously over here on the right is Alan Turing. Most of you might not know, but the Enigma in World War II was the Germans' code that they were using to communicate to the U-boats, which were sinking a lot of Allied ships. And Alan Turing and a few other scientists were really kind of spearheaded the effort in the cryptography of breaking that code. And he's standing next to this called the bomb, which was the British bomb, which was their kind of little device that did the kind of linear algebra and differential equations calculations. So, like I said, computers are used for calculating large numbers very fast, constantly. They also lend themselves to what's called stochastic models, which is the opposite of deterministic. Which also stochastic means it has some randomness in there. Some of the most famous methods are the Monte Carlo methods, which were used in early physics simulations for particles and what have you, diffusion of liquids, et cetera, et cetera. But also, which we'll kind of learn in the talk is Monte Carlo methods can also be applied to large data of sequence data, right? Large strings of DNA data. You can find hidden patterns in that data as well, which is pretty cool and kind of not intuitive. This right here on the left, I believe, is the Colossus, and this is a picture of the ENIAC, and then we got to have a picture of Claude Shannon on the right, which is he's the father of the digital age and really kind of spearheaded a lot of things in his essay. The history of the mathematical theory of communication, where he introduces information theory. So another big part of bioinformatics is statistical analysis, predictive analysis, this whole AI buzz, right? And what I kind of get frustrated with people is like AI this, AI that, but artificial intelligence is usually talking about some type of machine learning problem, right? Where you have data, you have a question, and you have to find an algorithm that will help answer that question or predict whatever you're looking to predict. And just off the top of my head, I just rattle off a bunch of these, which a lot of people kind of glaze over. You have neural nets, you have a bunch of other things. I just rattle these off just to show you guys kind of the extent of the really cool tools that you can use in bioinformatics and data analysis in general. And then finally, obviously, with a bunch of data and calculations and numbers comes the question of how are you going to store that and manipulate that and make sure it doesn't get washed away. All right, so moving on with the kind of history of computers in general, which is it's morbid. This is a picture of the atomic bomb, but the first program that the ENIAC, the electronical numerical integrator and computer, was a simulation of the atom bomb. Go figure. Then you have, I just wanted to mention the first commercial computer, which is the Universal Automatic Computer, which was the big kind of like buzz in the marketing on that was that it had tape where it could store things that weren't just numerical calculations. So it could be used for like text analysis or kind of census, like keeping track of the census of the population. And famously, it was used in the 1952 presidential election to predict the outcome. So now kind of going from, classical computer stuff to more biological segue into it. We got this article from Robert S. Ledley, which was published in 1965, which is the use of computers in biology and medicine. So this was like he was very forth thinking in the topics that he was discussing in this articles, basically highlighting that in the future, biologists would need computers you couldn't separate the two, right? So he was talking about whether it was calculating, systems, differential equations of systems describing, right, protein structure, blood flow, et cetera, or whether it was running simulations, biological system simulations, or which was interesting that he was talking about this in 1965, which was the, statistical analysis of like medical records, which you see a lot nowadays with the electronic health record. Like kind of Isal was talking about, he was doing some analysis. We were talking about earlier. He said he's doing some analysis himself, and without the EHR, without the electronic health record, that wouldn't be possible. So we're kind of in 1965. He had all these hopes and dreams for biologists and computers kind of meeting, which is kind of the definition of bioinformatics. It's like the bridge, the crossroad. But it didn't really come to fruition. The only thing that came about at this time was this thing called MoleGen, which basically was sequence analysis, like I referred to before, which is, you're comparing DNA, strings of really long DNA sequences, right, to find patterns or differences or similarities. And this is where these guys, Needleman and Wunsch, just remember that name. We'll get back to that in a second. They came up with the first kind of application of dynamic programming, which it just depends on where you're at in your training. You might have come across this, right? This is used to solve various algorithms, and it involves finding kind of an optimal solution. So, classic examples is like when you're coding the Fibonacci sequence or doing the Tower of Hanoi problem, which sure some of you remember. I'm trying to think right now. Um, I did an advanced algorithm class where we did it was the Farida. Have you heard of that one, Azad? Does that ring a bell?
No, I have
Farida.
not. I have not heard of that one.
So it's basically where you have monsters and you have coins and you have to figure out the optimal optimal pattern, how you take which coins you take from each monster, and it involves, n f of n, right? N minus one, n minus two, or in that case, plus one, plus two. Some recursion and some dynamic programming. So moving on, kind of get through this quickly. The Los Alamos, right, where they were studying, and a lot of researchers were packed in trying to, figure out the atom bomb. They had a bunch of specialized groups. In this case, the T10 group was a group of physicists that were very specialized, doing kind of simulations and things just physics related. But this guy Walter Goad, Goad, he kind of came in and he was a classical physicist. His training was trying to find patterns in the motion of neutrons and macromolecules. And like I said, he was using those Monte Carlo methods to find those patterns in that data. And he actually found that the DNA data, right? These long strings and just massive sequences were also akin to that kind of analysis. So he got interested in biology and started what was called at the time Los Alamos Sequence Database, but now it's referred to as GenBank because he got a bunch of funding from the government to start that. It's important to note that Margaret Dayhoff, it's another big name at this time. She was had her own research institute, but she failed to find the funding like Walter did, and she didn't get this big recognition of what GenBank is. And like I said, if you don't know GenBank, it's this huge institution. Izad, have you heard of GenBank?
No, I haven't. So I don't know much about bioinformatics other than the personal use that I've had in it.
Right,
So
right.
all these are
So
exciting new names that I'm hearing and history.
right. So GenBank is just this massive online sequence database where you can go get any sequence you want, whether it's human, animal, bacterial, viral, and it's maintained, and it's open access.
Is that the one that was created by this fellow? Is it now he has his boat in the ocean and is collecting all the DNA of the world, living things? I forget his name, starts with a V from Harvard.
Yes, that's no, no, it's not. It's not. But that dude is I can't remember his name at the
Yeah.
time. We have to look. You might have to look it up on that while you're listening. So then we get to the National Library of Medicine being interested in kind of, and I think this is around the 80s and the 90s. National Library of Medicine gets interested and starts funding things, including this thing called the NCBI, which is National Center for Biotechnical Information, which is also now like a huge institution. You can go to NCBI to find any bioinformatics needs. Pretty sure they have pretty much everything available, and also they offer training. So, Human Genome Project, everyone's heard of it. That was I could have led with that one, but wanted to kind of introduce some stuff that maybe you hadn't heard of before, and I hadn't heard of before I read this book called Life Out of Sequence. It's a really cool history of bioinformatics that my teacher in undergrad actually had us read. So Human Genome Project, everyone knows that it was the worldwide effort to sequence the human genome, and it's tens of hundreds of labs collaborating to sequence the entire human genome. And like this chart kind of shows you, it's around the time that the bioinformatics publications kind of go up in frequency. But around 1984, the term bioinformatics hadn't even really been coined yet. And it's actually not until 1994 that the Journal of Computational Biology was founded. And the book to the right, the Introduction to Computational Biology by Waterman, he's actually authored several other kind of bioinformatics algorithms. He actually authored the sequence alignment algorithm for proteins, which is called the Smith-Waterman um, algorithm. Or sorry, Waterman and yeah, so he's Waterman and Smith was his partner. Anyway, so this is considered the first bioinformatic textbook. I actually haven't checked this one out, but I wanted to after I did this kind of research. All right, so what is bioinformatics? It's a huge umbrella term. Um, Pretty much in my experience, bioinformatics is you can almost throw anything under there. Like I said, it's an umbrella term. Not going to go through every one of these, but right, it's programming, it's software development, it's data analysis, it's visualization, it's doing network analysis, it's managing databases, it's everything. It's natural language processing. It's pretty much everything that I find cool about computer science and stuff wrapped into one. And I'll tell you how that worked out, when I talk about my education in two slides. Second kind of tier of what it is things that you might not think about, right? Data stewardship, which is kind of how you manage data, how you treat it. Do you put the right security precautions on it? Because in healthcare, there's a lot of things called PHI, personal health information. So if you're managing a clinical study and you have all these people's genetic information, you really have to be a steward, a steward of that data. So obviously collaboration, things like I just mentioned, clinical trials, animal research, you can do it all bioinformatics. And then third tier is kind of, right, it's controversial. Before, I think it was 20 years ago, I don't have the exact reference pulled up, but it was, they came out with a study that said the death gene had been found, or I think this other one was like the obese gene had been found, and the New York Times picks it up and blah. So that's what the whole thing is like. It's controversial. Bioinformatics can be right there at the tip of the spear for breaking news and what have you. Another example of that is the BRCA1 gene, BRCA1. It's the breast cancer gene that was found. And Angelina Jolie, actually, the celebrity, she actually was tested positive for this, and then got both of her breasts removed because of a bioinformatics study. Justice, it's used in finding microsatellite regions in people's DNA to identify scenes of murder scenes based on blood or based on other bodily fluids with pretty brutal crimes. I remember the story. I'm not going to tell the whole thing, but it was a forensic chemist that came and gave a talk, and he worked on a naval base, and it was a family crime where there was some sexual assault and rape involved, and he did the bioinformatics analysis to figure out that it was not only the father but the son that had also been participating. So it was like it blew my mind, and I was like, wow, that is insane. So that's kind of why I put that third tier, because you know and he had to testify in court, right? So you can be a bioinformatician that specializes in that and have to go to court and testify for your analysis. Getting back into more happy happy vibe, talking about how did I learn bioinformatics. I did my undergrad at Whitworth, where basically it was the kind of beginnings of the program. Basically what the bioinformatics degree meant at that point was a quad degree of math, computer science, biology, and chemistry. So I had to take all the the good core classes and the 300 400 level classes of those. So you could imagine my final exam was pretty gnarly there. Then moved on to the post baccalaureate research at WSU Spokane, which was right there near Whitworth in Spokane, Washington. This is where it was like really cool. I got to really see what real research and what a real lab, not only just like a relaxed lab, but very high pace, kind of high pressure, like really cool kind of lab. That was a great experience. And then finally to Oregon Health Science University, where I continued to get my master's, and then was hired in the same lab that I did my master's in last. So I'm not going to go spend too much time on this slide. This is more for you guys, more for the people interested in bioinformatics or don't know if they're interested yet. This is a pretty complete kind of collection of what'll get your feet wet. just jumping into this type of field. This first book is just more about mathematical models. It starts with exponential population growth and gets into diffusion of fluids, et cetera, et cetera. But back to the first slide, right? It's systems of differential equations that describe some type of physical phenomena. Really good book. This is a really cool book. This dude Pavel Pesner is right at the head of kind of bioinformatics, revolutionizing kind of the way it's presented, like and this textbook is a testament to that. It's really good. More kind of classical machine learning textbook right here, the pattern recognition, the one in the middle. Uh, Really good. It breaks down all the calculus and has like proof by calculus in there. So if you haven't taken calculus, don't be too scared, but definitely try to go through at least Calc 1, Calc 2, 3, and differential equations to get the most out of this book. Then we have bioinformatics algorithms, another one, another kind of really good statistical analysis book. Natural language processing, which is a big part of bioinformatics, which maybe people might not think, right? Everyone's so concerned with ChatGPT and Grok and these large language models that you don't realize that those same type of natural language processing algorithms can be applied to things like the electronic medical health, the EMR, the electronic medical record, like I was saying before. You can do really cool analysis. Right. So this slide is just kind of a fun slide, right? So now the question is, what can you do with bioinformatics now that you learned it, or now that I learned it? And this is just like a hodgepodge of a lot of data visualizations that I've made over the years. And don't worry, it's all chill. Nothing's be able to. But boom, here we go. What can you do? Everything. Don't focus on every single chart, but the whole point of this slide is that you can make a bunch of really cool stuff with bioinformatics, stuff that I would have never dreamed of. Every analysis, every color you could imagine, right? So that's just the whole point of that stuff. And this is a really cool URL you can go to get practical experience doing bioinformatic problems. I should let's click on this. Okay, great. Yeah, so you can get the Python village, right, which will give you some Python. This is a bioinformatics stronghold. It'll take you through a bunch of really cool, kind of fundamental examples all the way through some more advanced ones. Like you can see, right, perfect example of transcribing DNA to RNA, right? So you have a string of DNA, you have to write an algorithm, you have to code it up in whatever language you choose and do it programmatically all the way through. You still good, Azad?
My microphone was off. You're still good. Go ahead.
Okay, sweet. No worries. But hey, that's a really cool link. People can go there, and even you, if you're interested, you can go there and start messing around because it's kind of fun. And that's what Pavel Pesner. That's what he. All right, cool. So now getting into my most exciting part of the talk, which sure is odd. Is looking at this slide like, what the heck? No, I'm just kidding. So many of you might recognize this comic book of Cerebro, which is Professor X wearing the mask. Some might recognize this one, and then others might recognize this one, depending on your age. But basically, this is my first taste of research when I was back in undergrad, and it was a really good example of some cool, like I said, computer science, foundational computer science mixed with biology. So the reason for the name Cerebro is because we were trying to genetically modify a enzyme to handle to help with celiac disease. So not going to get too into this chart. Don't be scared. It's it's biology, which I know a lot of computer scientists. Anytime they see biology, whoa, no, I'm just don't be frightened by the biology. I was once there, but then I embraced it, and now we're here. full on loving it. So the whole problem was you have the with people with celiac disease, your intestine is like this. It's very tight. If you have celiac disease, these toxic gluten fragments can slip through the intestinal epithelium. They can get kind of into this interstitial area, trigger the immune system, which might not ring any bells, but the interleukin 15, which is a part of the innate immune response, basically to deal with inflammation. And if you know inflammation, right, is bad no matter what it is. In this case, this is talking about the autoimmune response that it triggers that pretty much ends with these T cells attacking your own intestinal epithelial lining. So that's like the big deal with celiac disease. It's not good, and in some cases, some severe cases, even the slightest bit of gluten can actually cause people. It can be fatal for some. So celiac disease is no joke, and it's a big deal. So no wonder why my first project that I was a part of was inspired to kind of figure out some therapeutic for celiac disease. Now, the project was inspired by this paper out of Stanford, which took a bacteria from sourdough bread and was trying to modify it to make it more resistant to pH and heat, right? So it could survive in the gut and be able to help digest that toxic gluten in hopes of maybe, someone with celiac disease being able to take this as a pre-therapeutic and then kind of eat the gluten, and then the medicine will digest the gluten for you. So we took that as the inspiration, and like I said, the whole point is to modify a protein to help digest gluten better. Basically, that's at the end of the day what the gestalt of this was, and the goal is potentially to manufacture a therapeutic. So with these projects, I'm going to talk about three kind of things. I'm going to do biological health problem, and then what's the computational solution, and then basically why does it matter. So we'll kind of continue that same three themes for these two other projects that I'm going to talk about. So this is a visualization of the protein we chose was from, it's called Lactobacillus helveticus. It was from Swiss cheese. This is a visualization of that protein, and as you can see, there's different shapes in there, and that's how proteins are visualized. And a program, right, is behind this and visualizes it. So there's computer science behind this graphic simulation, this visualization, as well as some physics engine in there to determine the shape. So boiling this down to a quick, I'm not trying to get too far into this, but you have these different spots on the protein, and to manufacture or engineer this enzyme to be more resistant to pH and heat, we're trying to figure out which spots on this enzyme will do that, right, to make it stronger. And to get kind of so there was mutations made, and then there was some failures. So we were trying to figure out how these two parts were related to each other, if any. So before we talk about that, we got to talk about kind of a cool little interactive thing that we'll do. There's not many people to interact with now, but Azad's being a great participant so far. So we have to talk about multiple sequence alignment, which is kind of a cornerstone of bioinformatics. Have you ever heard of this, Azad?
Yes.
Okay, so this is kind of the cornerstone. This is like day one bioinformatics. This is a very common thing you'll need to do, which is compare sequences. This is one DNA, one piece of DNA. This is the other piece of DNA, and you have, these various sequences.
To me, it almost looks like a two-bit code.
Right,
You got
exactly.
four possibilities, so you got 2 bits to.
Yep, 4 possibilities, exactly. 4 possibilities, and you have 3 codons, so that's 64 possibilities for but we're not getting into that because that's more transcription. This is just the comparing of 2 sequences right here, which this actually isn't multiple. This is just sequence alignment. But anyway, so I mentioned Needleman and Wunsch earlier. This is their original article from the 70s talking about how they figured out how to compare sequences and figure out the optimal alignment. Because when you align two sequences, you're going to end up with one, right? So it's an alignment. Um, that's the first. That's the article, and like I said, they applied dynamic programming, and it was the first application of dynamic programming to a biological problem, as far as my knowledge. So this is pretty cool. So you basically go through. There's a scoring system. Every possible alignment is calculated. So here we go on the this part of the matrix right here. You have the first sequence. This part is the second sequence. You basically calculate all the possible alignments, and then you go back through and do a traceback routine, which picks the max. And right here is kind of visualize how it progresses. You're adding on the left. So, right, you're adding to the left. So here we go, right? GA, boom, pick the max one, CC, perfect. Here we go. Good, we're still good. That's the max. Now this is where you get a gap. You go up, and you can kind of continue and follow the rest as it builds up. Not getting into the algorithm, but it's pretty cool and it's nice. Pretty rudimentary because Smith and Waterman, like I mentioned, the guy who authored the first textbook, he improved upon it later. But still pretty cool. So that's just one sequence, right? And you get this alignment at the bottom. So multiple sequence alignment is a little more involved, where you have to do that kind of pairwise with, say you have 1,000 sequence, you have to do a pairwise, and then you have a nearest neighbor joining technique, which is kind of cool. It's kind of some graph stuff is there as well, but you can look up this algorithm, and if you want to dive deeper into that. The next question was, with that multiple sequence alignment, this is kind of how it looks. You have the first sequence on the top, and then you have, in this case, which was 1,000 other sequences next to it. These are each of those spots on the protein, like I was talking earlier, and they can be either what's called evolutionary conserved or variable. So conserved means that this is going to be a cysteine, no matter what, in all the 1,000 proteins that are similar to this one protein, and this would be the protein of interest. Like I said, this is a common problem. We didn't make this up. We got this from literature, and we were kind of just replicating what other people had done. So you can look at each of these spots and say you want to figure out a spot that's very related or has some statistical correlation involved. You can pairwise each column. So this column to this column, this column, this column. You could do a Pearson, right? You can do anything you could think of, and this is the case of mutual information. That's why we had the Claude Shannon and his manuscript there. This is a case of mutual information where each of these columns, big X, big Y, is a column, and you're doing pairwise on the probabilities. What I'm not showing is there's a matrix that shows you the probabilities of these two amino acids being next to each other. So that was kind of convenient because that was the probability portion that we used, but not focusing too much on that. Basically, you use that to figure out the values, and you end up with potential mutation sites based on statistical analysis to where you could genetically modify this enzyme to be more susceptible to heat and pH. So why does it matter? Well, because there's millions of people worldwide that have celiac disease, and this could potentially identify, this could potentially, like I said, act as a pre-therapeutic. So those people could enjoy sourdough bread and pizza and what have you. Obviously improve the quality of life. And, I think now is odd. There are some commercially available products for exactly this application. But back when we were doing this, it wasn't available yet. So the next project I want to talk about, try to move a little quicker since time's running out, but it's recorded, and hey, it's all good. Is you're good? Okay, sweet. So the next project I want to talk about is kind of like it's the title might scare you away, but it's called transcriptomic changes following sleep deprivation and recovery sleep. Transcriptomic changes following sleep deprivation and recovery sleep. Excuse me. So this is a hot topic, right? Everyone's worried about sleep deprivation, especially in college, especially if you're, you know, airplane pilot, you're in the military driving a tank, you're a law enforcement officer, working shifts, right? You're working the night shift, you're working the graveyard shift. You're a medical professional, you're a nurse, you're a doctor, you're a truck driver. Sleep deprivation is, I mean, it's as hot as topic as you can get, and as you could imagine, there's a lot of funding associated with this as well, right? Um, a lot of people want to know about this. So where I was at was the Washington State University Sleep and Performance Research Center, and the big question was, how does sleep deprivation affect the brain, and also asking the question of what is the function of sleep in general. I just kind of wanted to talk about in my kind of training and my experience meeting these two guys, Fred Turk and Greg Blanke, was like kind of meeting some like right, science celebrities. Really got me excited about research and what you can do. Because I mean, look at this is cited by right 4,000, and this guy's cited by 3,000 on some of his articles. Fred discovered what's called the clock gene in mice, which is the circadian rhythm. It governs your sleep and wake pattern. So he discovered that there was actually a gene that you could knock out in the animal, in the mice, to totally throw off its sleep wake pattern. You've heard of circadian rhythm, right, Assad?
Yes.
So he was kind of the forefront of that, Fred Turk, and then Greg Blanke. He's just like a legend for out of Spokane, and has just done a lot of really cool sleep research. And I wanted to mention that he told me that his first study, which was January 1968, his first publication, was keeping this cat, like doing the experiment how long he could keep a cat awake. And if I'm remembering correctly, he said it was like months or something. Like the cat would, it was as long as you kept feeding it water, and it was the coolest story I heard. I was like, wow, that's insane. Um, so getting back to kind of this sleep performance read and working at this project, right, which had to do with sleep deprivation, there's a big effort to figure out what's the function of sleep in general, whether it's for rest and recovery, whether it's to solidify learning, whether it's for survival, for simulation, right, because that's a leading theory of sleep, because you dream and the prey simulates the predator attacking it, therefore it can deal with that easier when it comes in the real world. So it's kind of this hallucinated enigma. And apologies for the slightly graphic image. We have a mouse that is getting brain surgery. That's how you figure this out. Okay, you go right to the mouse brain, you do some type of behavioral simulation, and you unfortunately have to sacrifice the mouse, and then you do gene sequencing on the different regions of the brain of these mice. So, like I mentioned, this is your normal kind of healthy sleep pattern, process C. When you have process S, which is kind of like sleep deprivation of sleep pressure, you're not able, right? You miss a session of sleep, you're not able to function, boom, and you collapse. This is like a classic graph, and kind of how you solve, right? What's the computational solution. So how do you solve this sleep deprivation problem? You have to use computational neuron science, like I mentioned before, which involves doing, brain surgery on mice and rats. So the way this experiment was set up, I actually went back and found some of these original graphs, which was cool. So you have a group of animals that are sleeping normally, and then you have a group that have sleep deprivation, and you have ones that have rest and recovery sleep. We're not going to focus too much on that, but basically you have three groups: one that's sleeping, one that's sleep deprived, and one that has rest and recovery sleep. So the question is, how do you do that with the technology and bioinformatics? Well, it's through DNA sequencing, like I mentioned, which you sequence the different regions of the brain. And I believe the regions for the mice were the hypothalamus, the thalamus, the cortex, and the neocortex. So you're doing this experiment, right, which is sleep depriving these rats, and then you're sequencing these different regions of their brain to see what genes are activated. Sounds pretty cool, right?
Yeah. The gene is amazing. I mean,
yeah,
it's been, Human health is going to benefit tremendously from us
Right.
understanding what the genes are doing and
Right.
all
And
the
in this case,
translocations and all
yep.
that.
In this case, it's the genes associated with sleep deprivation
Yeah.
and recovery sleep, which right is a pretty important topic.
I read an
Um,
article about a third of people in the United States have sleep disorder.
100%. Yeah. No, it's that's why there's all the money and the funding for these big centers where these the PIs I worked with, they're still going, and it's really cool. But obviously, getting back to kind of the theme of this talk, which is right, how did these computer science or math topics connect with the biology? Right, you can't get away uh, from statistical analysis, and in this case, differential expression analysis, which Azad just mentioned with translocation, how genes are differentially expressed. That's your classic, you know, baseline study, which you want to know whether you're a healthy person or you're a sick person, what the difference in the genes. And this is a specific gene that's associated with sleep deprivation, and you can tell that the sleep deprived group, right, it's more expressed in the form of these Boston Lister plots than the other groups. So that was just an example from that I pulled from one of the old presentations. And what you wouldn't know is that parallel programming and web dev are also associated with this project. So, things that you guys might be learning in your training, you never knew that it could be applied to a kind of a neuroscience application. And in the case of parallel programming, I optimized one of the scientist's scripts. He was using MATLAB, and I took the parallel library of MATLAB and optimized some of his code. And then the web dev, I made a website where all the scientists could access these types of visualizations, which is kind of sweet to use that training. Like I said, bioinformatics is a very umbrella. It's a very umbrella field, right? It's very versatile, and there's lots of things that lots of tools and skills that you might not think will be applied, but they come in handy. So the biggest point of this study is help solve this big question, this enigma: what is the function of sleep? As Azad mentioned, there's countless people all over the world that are suffering from sleep deprivation. So understanding the functions and everything around sleep is really important. We'll get crucial insights into our shift workers, which some of these are some of our most important workers, right? The cops that are working from noon to 12 a.m. to 8 a.m. The nurses that are going to be there when you go to the emergency room late at night. Really important. And like I said, it doesn't surprise me why those organizations would provide so much funding to figure this out, to figure this topic out, of sleep deprivation and sleep in general. And like we mentioned earlier, identify the genes involved in sleep deprivation and recovery sleep. So this slide should wake you up if you've fallen asleep, but hopefully you're still with me. This is talking about the work I did at the Oregon State Public Health Lab, which was a really cool experience. Might be wondering, how does Jack in the Box and Cold Stone, and strawberries apply to bioinformatics? Well, get ready to find out if you can't. All right, Izad, do you have any guesses? Oh, you're muted.
Guessing because you got all the food and it's ice cream that I'm guessing has to do with something to do with the strawberry and the ice strawberry used for the icing on the cake or on the ice cream or
What about
whatever.
Jack in the Box, though? Think it's all connected?
I don't know. There was so unfortunately the only thing I can remember about Jack in the Box is they had some sort of a problem with the
Yeah,
food.
the E. coli.
Yeah, E. coli. Yeah.
Yep. So hey, there you go. You hit it on the head. So this is basically talking about bacteria and how it's tracked all the way throughout the United States by the CDC and each of the individual state public health labs. So you have this program called PulseNet and FoodNet that tracks certain bacterias all around the nation. Say if you get sick in Montana at whatever it is, Jack in the Box or wherever you got sick at the cookout, you'll if you're one that goes to the hospital, you'll go to the hospital, they'll get a sample, they'll swab, and they'll culture out your specific, what's called a serotype, which is like a species, very specific species of bacteria. And then they'll send it to, a lab if they have the lab in that state. So then they'll send it to the lab, they'll culture it, and then they'll end up sequencing it. So the five or six that they watch is Salmonella, E. coli, Listeria, Campylobacter, and Shigella. I think I'm missing a few, but Vibrio and Corynebacter. But either way, these are all bacteria that can seriously mess you up. If you get any of them, it's bad news. If you get E. coli 0157, it can be fatal. So it's really cool to be a part of this, and obviously epidemiology, which is kind of the tracing of disease and how it starts, is that's who you work with specifically. If you're a bioinformatist, you worked with the epidemiologists, and also the microbiologists work with the epidemiologists as well. And just for funsies, we can check this out, this is the Bill. So William Keene, he was a really famous epidemiologist who worked in Portland. And as you can see, this is a lot of the stuff that they traced. Let's see, outbreak exhibits. This is outbreaks. Okay, perfect. So here we go. Jack in the Box, E. coli 0157, 93. That's the one Izad's talking about right there. They've got the exhibit for it. So they've got the cardboard and the whole thing. I think they had a piece of, no, I don't think they keep the samples, but anyways, where did the Cold Stone go? Cold Stone right here, 2005, Salmonella typhimurium. That's another really bad bacteria you don't want to get, and that's a multi-state outbreak. So working with these epidemiologists and doing this was really, really cool. I didn't think that bioinformatics would take me to that point of following, being able to trace disease in hold on, sorry. There we go. I never thought bioinformatics would take me to the point of, being able to prevent illnesses from bacteria that are growing in people's food. So I thought that was really, really cool, and like I said, even increased my my excitement for kind of what I was getting into with my career. So quick, going a little long on time, but I'll try to go quicker on these ones. So basically, kind of as I described, this is how the one of the computational solutions, which is this thing called DNA-seq. It's a really miraculous technology that involves the sequencing of an entire genome. Start with the bacteria, like I said before. You extract the DNA, you go through the process of cutting it, you amplify it in this thing called polymerase chain reaction. Big part of basically making sure there's enough samples so we can detect it. Sending it to the sequencer, which these are extremely expensive devices, right? Tens of thousands of dollars. They work with microfluidics. There's a whole bunch of physics that goes into these devices. At the end of the day, it's basically fluorescent dyes that tell us which base pairs are there. As you can see, the C's, T's, and G's. And from those fluorescent dyes, there's microimaging that goes on to give you the signal for what the string, right? This string of DNA comes out as. It's pretty cool. A lot of people much smarter than me were involved in making these machines, so it's really cool that I got to use them and set them up, which I did have a hand in that at the Oregon State Public Health Library. It's pretty cool. So you have obviously algorithms associated with that. Imagine a whole bunch of 150, 250 length sequences from the sequencer. You have to somehow put these all back together, and there's plenty of algorithms that do that. It's called whole genome sequencing. The one that I used for this project was called SPADES. And funny, it's that same guy, Pavel Pesner, his name there, which he was also the author of that textbook. He's right there at the tip of the bioinformatics education and research sphere. Again, There's other pieces to this. I actually just put my workflow. So, if someone watches this later, you can check it out. It's the workflow that you do to get from that biolog, from that culture, right? From this culture all the way through the sequencer to this point of having a sequence and being able to compare different samples and build this phylogenetic tree right here. So in this case, right, you have the hazelnut here. There's actually analysis I did. Hazelnut here. You have the culture from the people that are sick, and you can see that they're pretty close in evolutionary length, which is measured in these things called single nucleotide polymorphisms, which is just one change in the DNA code. So it's actually pretty interesting and pretty cool the things you can do. And those types of analyses would go towards the epidemiologists, and then they would say go out to that person's house or to that Jack in the Box and follow up and shut it down. Again, I wanted to include this in here because it's pretty cool. It's a piece of your classical computer science, which is setting up a headless compute node somewhere and having to use the command line to whatever it is, SSH into it and mess around and do some things, do some calculations. To do the required computation took a lot of compute power for this specific workflow, which is common for when dealing with this high throughput sequence data because it's right just millions of strings. So you could imagine you need a lot of compute power. We had a Google, it's just a compute node up in the cloud. This was Google Cloud specifically, and right, this is just like a tight VNC window, just showing you how you can tunnel in and even have a GUI on a headless machine. So pretty sweet. The stuff I'm saying might not make sense. I know it does to you, Azad, but maybe to someone that's a freshman or sophomore. But trust me, the command line is going to follow you wherever you go. So get used to it. And I think developing your skill set is really important in that because that was not intuitive to me either when I got into bioinformatics. Why does it matter? Kind of already went over this, right? Safety of millions of people, identifying these bacteria that are potentially deadly, right? Shut down the restaurants and the farms, the producers to figure out the source, cut it out, go on a recall, and then move on from there. And then you can also discover brand new bacteria that maybe have been brought from a different country or a foreign, or origin from a reptile or something like that. Because the lab that I worked in, I think it was the lab, the sister lab, they thought they found a new bacteria, but it was because a man had a bunch of reptiles at his house and he was kissing them. So he actually got salmonella from lizards because they naturally have it occurring in their mouth. So it's pretty funny, the kind of anecdotes you can get doing this. So this is the last project I'm going to talk about. And it's the one I did at OHSU, which is elucidating gut microbiota immune interactions in an animal model of ankylosing spondylitis. So that's a mouthful, but basically what ankylosing spondylitis is, it's arthritis of the spine, and it's an autoimmune disorder that kind of causes a lot of pain and discomfort in the worst cases. In the more milder cases, people can be fine living their whole lives with AS, but it's an autoimmune disorder, and it's thought that the gut microbiome has a lot to do with that inflammation, and there's a connection between the gut microbiome and joint inflammation because there's a lot of shared similarities in the inflammation that you see in the gut and the inflammation that these people have in their joints. So that ankylosing spondylitis autoimmune disorder is caused by what's called the HLA-B27 mutation. It's a mutation that's in the HLA gene region, and it's pretty common. In this case, this is actually the strongest rat model, transgenic rat model that's been made or that's been studied. Why it's transgenic is because there's actually human copies of the HLA-B27 gene in these rats. So it's pretty interesting, and I didn't know that going into it that you could do that. What's cool about these, or not so cool about these rats, is that they exhibit the same symptoms that the patients do, which is enlarged ankles, uh, not solid feces. So they're having kind of diarrhea, which is a lot of uh, things you see in the humans as well. So basically, the whole goal of this project is comparing healthy rats to these HLA-B27 rats to try to figure out some link to the human disease, so we can identify bacteria that are causing this inflammation. And like I said, so it's a link between intestinal dysbiosis, which dysbiosis just means there's a dysregulation going on. Normally, like I said, the rats are supposed to have solid poops and not have inflamed joints, but in this case, they have inflamed joints and runny poop, which indicates that there's something going on in their gut. So real quick, the workflow for this was you take the fecal pellets, you extract the bacteria, and then you stain with this what's called immunoglobulin A, which is immunoglobulins are a big part of your immune system, and immunoglobulin A specifically uh, actually adheres itself to the bacteria to label them for destruction in a healthy immune system. So then you get to use this really cool machine called the FACS, which is the fluorescent activated cell sorter. I mean, it's a couple million, I think it's couple $100,000 machine. It's very, really expensive machine, really cool to use. I didn't get to operate it myself, but one of my lab partners ran it. So you put that in there and it sorts out the bacteria that are stained by the IgA and the bacteria that aren't. And then after that, you do what's called 16S sequencing, which basically is sequencing the bacterial genomes that are present in that stool sample. And in the case of machine learning and predictive analytics, like I had mentioned before, this shows you the kind of cool machine learning problem that was posed for this. So basically, in machine learning, you can do a few things. You can do classification, regression, or you can do what's called like feature optimization or dimensionality reduction of features. But classification and regression are pretty much the two popular ones. In this case, I'm going to do a supervised binary classification, which means there's two groups, control and a case, right? And the algorithm knows which ones. So when you do a supervised binary classification, you have a training set and a test set. So in this case, the training set, the algorithm was trained, telling you what you can expect or the patterns you can see in the controls versus the cases. And then the whole thought was that eventually down the road, you could build this algorithm to be able to identify not only rats, but also identify the human signature of AS or of gut inflammation. So in this case, right, you have a matrix. All the features are the bacterial species. You have the values for them. In this case, I chose a random forest. And it's important to note, like if you chose a neural net here, you might be able to predict the outcomes very well, or complete this classification successfully, or with a high accuracy and everything. But you're not going to be able to open it up and look under the hood, because neural net is a black box algorithm. Where these ones, which you might be familiar with, logistic regression, linear discriminant analysis, and in this case, random forest, you can actually go back and find out what features, in this case, bacterial species, which bacteria were the ones that were important in predicting or determining between the healthy and the diseased animals. So why does it matter? Because the theory that the bacteria can translocate from the joints and get centralized and pushed all around to the peripheries is a novel hypothesis, and it's pretty much the leading one. You can identify which bacteria are the causing the inflammation, and you can target them, or in many cases, you can have a fecal transplant actually between a healthy person and someone that has dysbiosis or has an autoimmune disorder that is affecting their gut, and it can lead to potentially curing the disease, or at least providing some therapeutic intervention. So all in all, if you're still here with me, if you haven't turned it off, the big conclusions are that, right, if you have the computational skills, if you put in the work with your training, if you see it through, get your bachelor's of science or get your bachelor's in computer science, and maybe you want more, or maybe you want to you don't you're not ready to go in the workplace just yet, or you want to apply it to some other discipline. The computational skills will get you there. Um, I've worked on projects that have been right there on not necessarily cured cancer, but on the way towards it, right? And other debilitating diseases. Um, and in my case, bioinformatics really allowed me to apply, everything that I've learned, and really allowed me to work on some super cool multidisciplinary projects that, are priceless, and that gave me kind of the tools and skills to be successful later on in my career. So with that, I'd like to thank you guys, and probably don't have any time for questions, but
Sure. So thank you, Mark. Yeah, we don't have an audience today. This was more of a recording to be broadcast later on. My apologies for that. But thank you for a very informative. There was a lot of new things in your presentation, even though this is something that I've personally benefited from medicine that has been created because of data science. But there was a whole bunch of things that I wasn't aware of that you shared, and I'm sure our students will benefit from your position. So thank you for that, and coming over via Zoom and presenting the information and preparing this. Thank you very much.